So catabolic reaction is a breakdown of large molecules into simpler ones. So an example of that is in relation to it is the exergonic reaction, wherein we break down the glucose molecules to release ATP. And anabolic reaction is when we build molecules together. Okay, to produce a larger molecule. Okay, in line with that, anabolic reaction is also endergonic reactions, which requires energy for it to occur. So the next part is that we are going to explain the induced fit model and inhibition. The next area that is needed to be discussed under enzymes is about the activation energy. So we have to tell our students that enzymes are being used in cells because it lowers the activation energy. So what is an activation energy? An activation energy is the energy required to raise the substrate into transition state. So we have to make a review of what transition state is, and this has to be this needed to be discussed in the core part, wherein whenever the substrates are going to be raised in a transition state, this is a point at which the substrate, together with the enzyme, the substrate is going to be changed into a product. Okay, so this is the point wherein the chemical bonds that is present within the molecule of the substrate is going to be broken down. Hence, this is actually placing the substrate into, or making the substrate to be changed into a product. Okay, so I have a graph here showing the reaction coordinate between the substrate and being changed into a product. So as you can see here, the ones that we have lined in red is the chemical reaction of a substrate being changed into a product without the use of an enzyme. So as you can see here, we have used too much activation energy just to change the substrate into a product without the use of an enzyme. Whereas for the green one, okay, the green here, the green mark is being represented by the chemical reaction being used whenever a substrate is going to be changed to a product, being facilitated with the use of an enzyme. So as you can see here, we only use a very minimal amount of activation energy. So whenever our cells use enzymes to change the substrate into a product, therefore we actually save a lot of free energy for the product, which is going to be used by other enzymes or by other chemical or metabolic processes along the way. So by explaining what activation energy is, the students would have a knowledge of what their importance is. So this is how enzymes are being effective in the chemical reactions that are occurring in cells. Okay, previously in the core part of enzymes, we have discussed that for enzymes or for substrates to be changed into products, they have to bind to a specific enzyme. So for every substrate, there is a specific enzyme that is actually working with it. So that is actually made possible because of the active sites. And active sites, they have specific amino acid residue group that is actually matching with the substrate that enables it to be changed into a product. So that in that case, this identifies the specificity of an enzyme, that an enzyme works specifically onto a specific substrate because of the amino acid residues that is matching together with the enzymes in the substrate. So we can actually relate that explanation with lock and key hypothesis. Since we are now discussing the more advanced or more detailed part of enzymes in HL students, we have to point out that enzymes does not bind rigidly with the substrate, or substrate does not bind rigidly with the enzymes. Okay, because the reason why enzymes are very effective in metabolism and in chemical reactions, because they are continually being reshaped, and they are continually being changed momentarily for them to facilitate their function, which is to help or aid in the chemical reaction occurring in cells. So for that, we are actually using the induced fit model for the specificity of enzymes, wherein we can actually explain induced fit model by just simply saying it actually works like a glove. Whenever a glove is being fit onto into a hand, then the glove is actually perfectly shaping the glove is actually being perfectly fit with the hand so that it will be able to work. In this same case, the enzyme is working effectively with the substrate because there are specific amino acid residues that are molded at precise areas or precise points that enables the enzyme to perform its function. Okay? So whenever that happens, for example, we have an enzyme here, and then this is our active site. Okay, provided that this is okay, so this is our enzyme, and then this is the active site of the enzyme. Okay, so here the active site of the enzyme, there are amino acid residues here that should match with the substrate. Okay, so when that happens, so if in case that the substrate binds onto, okay, let's just say I'm gonna draw here. Okay, so for example, this is the substrate, okay, binding onto the active site. There should be points here, there should be amino acid residues here that should match. And these amino acid residues are continually being reshaped and moved along the way so that whenever this amino acid residues match all together with the substrate, the bonds that is holding the substrate molecule all together is going to be broken down. So when the bonds are being broken down, it facilitates the changing of the substrate into a product. Okay, so that explains the specificity of enzymes being explained through induced fit model. So the last area that is needed to be discussed under enzymes is about inhibition. So inhibition, this is a process wherein it lowers or it decreases the rate of reaction of the enzymes, simply because there's the presence of inhibitors. Inhibitors, okay, they are molecules or they are substances that are able to bind onto the active site of the enzyme, or they bind at a different site within the enzyme molecule that prevents the or which actually changes the shape of the active site of the enzyme. So when that happens, this prevents the substrate from binding onto the active site, or actually if the shape of the active site has already been changed, then it also prevents the substrate in binding on that, because they don't the substrate and the active site doesn't match. So we have two types of inhibition, and we have to discuss their differences. The most common question that they usually get, that students should get in this type of topic, is to differentiate competitive and non-competitive inhibition. So here, in the case of competitive inhibition, the reason why it's called competitive, because the inhibitor competes with the substrate for the active site. So the structure of the substrate and inhibitor are similar. So they are structurally similar to each other, or they are chemically similar in a sense that they share the same chemical groups or molecules molecules that is present in that structure. So when that happens, the inhibitor occupies on the active site. So whenever the inhibitor occupies on the active site, this prevents the substrate from binding with the active site because the inhibitor is there. So how can we overcome this this situation? We can only overcome this situation whenever there is an increase in the substrate concentration. By increasing this concentration of the substrate, then we are able to displace the inhibitor from the active site, and then allow the substrate to bind with the active site. In the case of non-competitive inhibition, this is when the inhibitor itself is not chemically similar with the substrate. So the inhibitor is actually binding at the other site or other area within the enzyme molecule. And when that happens, whenever this inhibitor binds on the other side of the enzyme molecule, this changes the shape of the active site. Of course, whenever there's a change in the shape of the active site, this also prevents the substrate in binding with the active site. So we cannot actually reverse this chemical reaction. In the case of competitive inhibition, this is a reversible reaction. Non-competitive inhibition is irreversible. So sometimes they would ask to give examples of non-competitive inhibition, an example of which is allosteric inhibition. So when you say allosteric inhibition, this is when there is a metabolic pathway, and along that pathway there are some areas that is being switched off. So switched off metabolic pathway. Okay, so it can be switched off because of the increase in the concentration of the product, which affects the first stage of chemical reaction, which is the first enzyme. So we can actually give an example. The example that we can give for the allosteric inhibition is end product inhibition. So in the case of end product inhibition, so for example, we have substrate A, substrate B, substrate C, and then substrate D. Okay, so substrate A will be changed into B using enzyme A, B will be changed into C using enzyme B, and C into D using enzyme D. Okay, so when this happens, okay, of course this is actually a pathway, a chain. Okay, so whenever there is an increase, okay, let's just say that the substrate A will be changed into B, and then B into C, C into D. Okay, being changed by their appropriate enzymes. So here, at the end of the process, there is an increase in the concentration of substrate D. When this happens, whenever the substrate D increases in concentration, this could affect the activity of enzyme A, because the increase in the concentration of substrate D prevents or switches off the activity or inhibits the activity of enzyme A. So when that happens, we can only overcome this whenever there is a decrease in concentration of enzyme D. If enzyme D decreases in concentration, then it can go back to its normal cycle or normal state, wherein enzyme A will again work effectively, but hence later on increases the concentration of substrate D. So that will be all for enzymes and nucleic acids and proteins.